prokaryotes, the number of genes that belong to a specific functional class scales with the total number of genes, and this scaling can be approximated by a power law, and this power law can have a scaling exponent that can be sublinear, superlinear, or near linear. So, for example, if we look at translation genes, the number in the genome is approximately constant. The scaling exponent is very low, 0.35 in this case. Contrary to that, if you look at transcription genes, they scale almost quadratically. In this case, the scaling exponent is 1.63. And the question is why these different scaling exponents are observed. What is the underlying mechanism of this scaling? Now, this is something which is universal and is observed for any classification of genes. So we checked it for 20 functional categories of the COG database, and in our analysis we looked at nearly 1,500 genomes from the ATGC database. Now, the scaling is not new. It was originally reported back in 2003 by Van Inwegen and in various more publications. So one of the leading explanations from this scaling phenomena is that there are basically three integer exponents, 0, 1, or 2. And whenever we observe something which is in between, it's because either we mix different categories or because of classification ambiguities. And the explanation for 0, 1, or 2 are quite intuitive. So the exponent of 0 means that the number of genes is constant, namely translation genes. There is a certain amount of translation genes that are required in the genome, so therefore the number is constant. The exponent of 1 indicates a linear growth, for example, metabolic genes. And the exponent of 2, which is kind of puzzling, is explained by the toolbox model. For example, for regulatory genes, the number will increase quadratically with genome size because the existing building blocks can be incorporated in new regulatory circuits. So the toolbox model addresses the quadratic scaling. And now we want to analyze and understand what's going on. So first of all, we see that actually it is not, the scaling is not a power law, it's something which is a bit different. And in fact, we can get any kind of scaling, not only integer scalings, as I will show. So the scaling is just counting the number of genes. We want to look at complementary ingredient that has to do with genome content, which is the genome pairwise intersection. So whenever we look at a pair of genomes, always there will be a certain amount of genes which is shared by these genomes and some genes that are unique to each one of the genomes. In technical terms, we can describe the genome as an array of 0 and 1 representing presence absence of genes. In this context, I will also define cogenome and pangenome for later use. So when we have more than 2 genomes in the cluster, it makes sense to define the cogenome, which is the number of genes that are shared by all genomes in the cluster, and the pangenome, which is the non-redundant collection of all genes from all genomes. So in this plot, any point represents a pair of genomes. And we see that the number of genes, the intersection, the number of genes that are shared by a pair of genomes decay with 3 distance. I will show that as a first approximation, this is exponential decay with 3 distance. This was previously observed for archaea, bacteria, and bacteriophages. So this kind of decay is something which is general. And now we want to tie everything together and understand how the scaling laws are related to the intersections. For that, we need a model. So the evolution of prokaryotes is subject to extensive gene loss and horizontal gene transfer. So the simplest model to depict the evolution is a succession of stochastic gains or losses, which are governed by gain rate and gain loss. So gain rate and gain loss incorporate the mutation frequency, which in this case is acquisition or deletion, and the selective effect. Now, note that in this kind of setting, there is a symmetry in the selective effect between gains and losses. In fact, there is a short-term phase for an acquisition, which is known as the AGT barrier. This is incorporated and hidden here in the gain rate through the acquisition rate, which is incorporated there. Now that we have the model, we can also calculate the decay of the pairwise intersection. So basically, if we assume that there is an external infinite gene pool, we get that the intersection decays exponentially when the decay constant is given by is proportional to the per gene loss rate. So the important point here is that the genome intersections are dominated by the loss rate and are not do not depend on the gain rate. So now we have everything in hand, and we can come back and analyze actually a subgroup of genes. So we apply the model for class-specific genes. So X1 is the number of genes, and P1 plus, P1 minus are gain and loss rate. We assume an equilibrium, we get the scaling, and then assuming that the horizontal gene transfer barrier is similar for all kinds of genes, we get that the scaling is only due to the selection landscape. So sublinear scaling is associated with a selection landscape that decreases with genome size, and superlinear scaling is associated with a selection landscape that increases with genome size. However, we have additional piece of information, which are the genome intersections, and we actually see that only a selection landscape that decreases with genome size is compatible with the genome intersection. So only panel A is relevant, and in fact the superlinear scaling selection landscape, which is increasing, is not compatible with genome intersections. And the reason is that the horizontal gene transfer barrier is not the same across all types of genes. And therefore we call it genome plasticity. It reflects the ability of different circuits or pathways to accommodate or to tolerate additional genetic material in the short term. And specifically, the selection landscape dominates the loss rate, and genome plasticity is the major determinant of the gain rate. In technical terms, we can infer the selection landscape for the decay constant of the pairwise intersections, and then we incorporate also the number of genes and get genome plasticity. We calculate the genome plasticity for all 20 categories, functional categories of the COG database, and in fact, we see that genome plasticity is correlated with the scaling exponent, and also the genome plasticity also is correlated with the pangenome and the cogenome, because it is a measure of how easy for the circuits is to accommodate additional genetic material, and everything that I described is now available in the print in bioRxiv. And if you're interested in hearing more, please come by my poster later today, and thanks a lot for your attention.
Yeah, any questions? Any questions?
Okay,
Oh, yeah, okay, yeah.
So you said that the number of orthologs decreases with the distance?
Yes.
Do you think this might be confounded by our ability to detect these orthologs? Or?
No, this is something which is, so basically, if I understand the question, is because they are too divergent based on sequence similarity. So as you can see, the tree distance here is quite modest. So I think it is possible to detect them quite accurately. Also, in other studies, for example, in bacteria, they studied something slightly different. The distance was measured using the 16S, and they observed the same. Okay. Thank you.
Okay, thank you. Yeah, now, yeah, we have a change. Yeah.
So I'm the new chair for you this morning.